Okay, hi everyone. So I'm Elaine Lim, and I'm an assistant professor at UMass Chan Medical School, having started my independent lab about 3 years ago after finishing up my postdoc with George Church at Harvard Medical School. So today I'm really excited to talk about some of the initial single-cell spatial RNA sequencing that we've been doing together with Complete Genomics using their Stereo-seq technology to try to understand the impact and role of viruses, such as herpes viruses in Alzheimer's disease, with the use of donor-derived brain organoids or cerebral organoids. So we started with iPS cells, or induced pluripotent stem cells, from a somewhat de-identified donor codenamed PGP1. We differentiated these iPS cells into three-dimensional cerebral organoids for 2 to 4 months, and subsequently we dissociated these cells into what we call 2D dissociated cells for viral infections, followed by bulk RNA sequencing. So we have a manuscript currently that we are preparing to demonstrate that actually HSV1 infection in these cerebral organoids do result in ADE-associated molecular and cellular pathologies. And we are using our systems to discover novel therapeutics that could be used potentially to treat a subset of patients with Alzheimer's disease. So in parallel, we took some of these 2D dissociated cells and performed infection, and we ran this using PASS Biosciences technology to look at single-cell, non-spatial RNA-seq data. And in collaboration with the Complete Genomics team, we took some of these three-dimensional undissociated organoids for viral infection, and we embedded them into cryoblocks, followed by sequencing with the Stereo-seq platform. So the organoids are pretty small, about only 3 millimeters in size. So for a 1 centimeter by 1 centimeter cryoblock, we actually embedded 6 organoids in each cryoblock. And you can kind of see the spatial map from one of our slices that you can kind of see the 6 organoids over there. So why were we doing this other than just generating a lot of transcriptomics data sets? So we are very interested in trying to understand, the role of common viruses, starting with herpes viruses, but eventually SARS-CoV-2 as well, in potentially causing neurodegenerative diseases such as Alzheimer's disease. So in our initial work, when we looked at viral infections of HSV1, one of the herpes viruses, in these 2D dissociated organoid cells, we found that we got very good infections. In fact, you can see the microscope image, the virus has a GFP tag, and we got and we ran flow cytometry on these infected cells, and typically we observe, something like 30% to 80% of cells were infected by these viruses when in a 2D state. However, when we took these 3D organoids to do similar viral infections, no matter how much viruses we actually bathe these organoids in, this was kind of like the microscope image that we got most of the time, that relatively few amounts of cells could be infected by the virus in a three-dimensional state. And in fact, most of the infected cells were just on the periphery of the organoids. And we did flow cytometry as well and confirmed that we got very low rates of infected cells, somewhere around 5 to 15% at most. So we were wondering, right, given that herpes viruses are very common in the population, about 60% of us might have been infected with some herpes viruses at some point in time. However, not all of us would go on to develop Alzheimer's disease. So perhaps studying the 2D versus 3D, uninfected organoids could reveal insights into what is being perturbed, both from the viral as well as the host transcriptomics. And perhaps that could lead us into understanding not just the basic biology, but what target genes to go after in terms of therapeutics. So looking at some of our initial results from using Stereo-seq, we mapped the expression of GFP, on the left, and we saw, the red dots, and we saw that, as expected, the infected cells were mostly on the periphery of the organoids. The red spots are just on the outside. And similarly, when we mapped the viral transcripts that we were able to get from the Stereo-seq data, we saw a very similar pattern, that it was mostly the peripheral cells that had a high amount of viral transcript expression. Now, taking a step back, when we look at our 2D data that was generated using PASS Biosciences, and I should mention that both the PASS and the Stereo-seq data were sequenced on the DNB-seq sequences. So when we look at the 2D data, the 2D organoid single-cell non-spatial data, we saw that there was very clear transcriptome-wide differences, looking at the host transcripts on the left and the viral transcripts on the right. So, the infected samples and the uninfected samples had massive transcriptome-wide differences from this 2D organoids. However, we didn't see that for the 3D, the Stereo-seq data. So looking at the clustering shows a better representation. For the past data, the 2D organoids, there was very clear clustering. All the infected samples were more similar, or the uninfected samples were more similar to each other. However, for the 3D, the pattern wasn't as clear. There wasn't massive transcriptome-wide differences between our uninfected and infected samples. We use a statistic that we developed called gene score to basically enable us to compare the single-cell RNA sequencing data that we generated with prior bulk RNA sequencing data that we had generated. So generally, if we see gene scores that were greater than 0, like high up, that represents that there is stronger concordance between the single-cell data with the bulk RNA-seq data. And the x-axis shows per cell type cluster. So for the past data, the 2D organoids, we didn't really see much inter-cluster differences. In fact, most of the clusters showed a very wide range of concordance or discordance with the bulk RNA-seq data. There wasn't much going on, or there wasn't much informative, sort of like cell type specific information that we could get out of this 2D single cell RNA sequencing. However, when we looked at the Stereo-seq data, the 3D, from the 3D organoids, we saw that there was huge differences. In fact, there was significant inter-cluster differences that we were able to, sort of pull out. In fact, one of the cell type clusters, like cluster 7 over here, showed a very high, discordance with the bulk RNA sequencing data. So we were really, really intrigued, and this is not so for some of the other cell type clusters that were out there that showed much higher concordance with the bulk RNA-seq data. So looking at the density map of the Stereo-seq data, our uninfected sample is on the left, the infected sample is on the right. We noticed that there was a particular cluster of cells that stood up as being more represented in the infected samples, that's boxed up in the red at the bottom. And this particular cluster was primarily comprised of cells that contributed to the signal in cluster 7. And as a reminder, cluster 7 was the cell type cluster that we saw very high discordance with what we had observed in the 2D organoids, especially using bulk RNA sequencing. So right now we're super excited. We're trying to dig a little deeper into both the viral expression as well as the human gene expression to try to understand what does the virus do that's different when it's infecting 2D organoids versus 3D organoids, and perhaps that could lead to novel insights into therapeutics. So as a summary, we are really excited about the use of single-cell spatial RNA-seq to try to gain novel insights into biology. For our purpose, we were looking for an unbiased way to sort of assay, these transcripts, at a single-cell level, but also with spatially informative resolution, and that's why we opted for RNA-seq, a sequencing-based platform instead of an imaging platform. And this allows us to generate hypotheses as well that we can go on to test. And the use of a sequencing-based platform was extremely important for us because we could then do data integration with other sequencing-based omics data sets that we generated in our lab, such as the bulk RNA sequencing or the spatial or the single-cell non-spatial RNA sequencing data sets. And as a future direction, our group is also working on calling single nucleotide polymorphisms because we're very interested in growing mosaic organoids comprised of cells from multiple donors to try to understand, the impact of viral infections in multiple individuals. And potentially as a longer-term future application, as single-cell long-read RNA sequencing technologies become more available, we are extremely interested in trying to identify alternatively spliced transcripts. Yep. So with that, we have a lot of collaborators today, including my postdoc mentor, George Church, David Knight, who's a HSV1 expert at Harvard Med, Tracy Young-Pierce is an expert with Alzheimer's disease, iPSC modeling, and I also like to thank the late Michael Green at UMass Chan, who helped to facilitate this collaboration with Stereo-seq. Thank you.